Okay, so this is chapter 9, which is about carbohydrates. So second to the last chapter natin to sa ating tutorial classes this semester. Okay, so sabihin malapit na tayo matapos. Okay, and then ang katugtog nito will be the chapter 10, metabolism. Okay, so you should watch this first before you proceed to the next chapter. However, sabi ko sa inyo last time, isang quiz lang ibibigay ko kasi pagsasamahin ko na yung coverage nilang dalawa. Okay, so yun. So let's talk about carbohydrates. No? So carbohydrates, alam natin that this is the energy source, no, for us. No? Kasi kapag tayo kumakain, um, lumalakas tayo. No? So kunwari gutom na gutom ka, nangihina ka. Pag kumain ka, nagiging energetic ka. Okay? Because you are eating carbohydrates. No? Okay? So ano ba yung carbohydrates? Okay? So, ang carbohydrate, this is the main source of energy for us. No? So, ito ay gawa sa carbon and the hydrate, which is H2O. Okay? So, ibig sabihin nun, ang formula nito ay CH2O. Okay? So, yan. So, by changing the number of atoms there, then you will produce carbohydrates, many types of carbohydrates. One of which is glucose, no? Which is yung ating primary source of energy sa ating cells, no? And then when glucose is being combusted in our body, they produce ATP, which is the energy for the cells, na. Okay? So, yun yung dahilan bakit lumalakas tayo when we eat carbohydrates, no? So, ano ba yung mga sources ng carbohydrates? Marami, no? So, generally, halaman yung nagpo-produce ng carbohydrates, no? So, hindi lang yung bigas na pinoproduce nila, hindi lang din yung prutas na pinoproduce nila, even the wood trunk itself and the dahon, gawa yan sa carbohydrates. Okay? Kaya nga mapapansin nyo, may mga animals na kinakain yung dahon, no? May mga insects na kinakain yung kahoy, kasi nadadigest nila yon. Okay? So, nadadigest nila yon. Kaya, yun, nagiging sugar siya na nagiging energy source ng mga animals and insects na yan. Okay? For us, hindi natin magagawa kumain ng kahoy. Una kasi, hindi naman talaga tayo design na kumain ng kahoy. No? Siguro imagine mo na lang yung mundo, no, kung pwede tayo kumain ng kahoy. E di walang world hunger. Kasi kukuha ka lang ng kahoy, kainin mo. No? But apparently, that's not the case. No? We lack enzyme that will allow us to eat wood. No? Yung mga may enzyme na yon, yung mga insekto, for example, yung mga anay. No? So, sila lang yung pwede makadigest ng kahoy. Actually, yung ano din, yung ipis, no? May enzyme sila to break down the cellulose sa kahoy para maging sugar. Okay? But for us humans, that's not possible. Okay? Yeah, when you eat fiber, no? Pag nilabas mo yun sa inyong ano, after some time, no? Pag nilabas mo yun sa body mo, buo pa rin siya. No? For example, yung pulp ng mais, pag kinain mo yun, ganun pa rin siya. No? Although, yes, gawa rin yun sa cellulose, but gawa yun sa sugar, but we are not designed to consume that. No? Hindi tayo designed para ubusin yung mga types ng products na yun. Kaya yun. Pero yun nga, no? So, dami ko sinabi. Carbohydrate, ang primary source natin dito is the plants, which is very abundant sa earth, no? Ayan. And whenever there's plant, then ibig sabihin may pagkain. Okay? So, yun. Also, pag carbohydrates, ang present na functional group natin dito is the hydroxyl groups, no? So, since it is composed primarily of hydroxyl groups, then it can interact with water. Okay? So, yan. Nag-i-interact siya with water. Kaya nga yung kahoy natin, it can pull water from ground up to the leaves, na kaya niya higupin yung mga water na yon from lupa papunta sa dahon. Kasi nga, its polarity due to the presence of OH functional groups in the sugar allows it, no, to interact with water. So, magpo-form siya ng hydrogen bonds, then papasa-pasa niya yung water until maka-reach yon sa dahon. So, ganun yung ginagawa ng ating mga they maximize the hydrogen bondings. Kaya, yan, nata-transport nila yung water up to the leaves. So ano yung purpose ng carbohydrates? Ano role nila sa buhay natin? Marami. No? So alam natin this is the primary source of energy for us. No? Humans, animals, insects. No? Lahat tayo halos gumagamit ng carbohydrates to produce energy. Also, sa previous discussion natin, alam natin that our DNA and RNA ay may sugar kasama doon. So, pwede deoxyribose, pwedeng ribose sugar. Okay? So, yun. So, depending on the type of sugar that we have, no, pwede tayo makakuha ng either DNA or RNA, which is very vital kasi dyan naka-store yung ating genetic materials. No? So, andyan yung instruction kung paano tayo mag-exist. Okay? So, another thing is that, to medyo hindi ito obvious, some bacteria, okay, have sugars in their cell wall. No? Alam natin kasi yung plants may cell wall in the form of cellulose, di ba? However, there are bacteria na meron siyang carbohydrates. Okay? So let me show you. Ayan. So ito yung ito yung energy source, ito yung ating DNA, RNA. Papansin mo may sugars ka doon. Okay? So it links the phosphate and the nitrogenous bases. Ito naman itong bacteria natin. So may types ng bacteria na mayroong cell wall na sugar, no. So yung tawag sa cell wall na yon ay peptidoglycan layers, no. Okay. So yan. Yan ay common sa mga gram-positive and gram-negative bacteria. Okay? So yan. So meron silang sugar sa coating nila. Okay? And other than that, when our sugars are connected with other molecules such as lipid, no, they serve as parts or members of our cell recognition processes, no. Okay, so usually nasa labas yan ng cell membrane natin, mga naka-point outwards, di ba? You have the lipid bilayer, then sabi ko may mga nakatusok palabas doon. Okay, so yung mga nakatusok palabas na yon, sometimes they are sugar bonded with lipids, no, and they serve as the receptor site for the cells, no. Doon kumakapit yung mga viruses. Okay. So doon sila kumakapit, or yung mga gamot doon din kumakapit, no. Okay. So yan. So they are used to form the recognition sites for hormones, antigen, and etc. Okay. So ngayon, let's look at the structure of sugar. May illustration na pala ako dito. So these are the typical structures for sugar. You have this image here na naka-straight line lang siya. Characteristic dito yung aldehyde functional group. Then you have the hydroxyl functional groups here. Okay. And then you have your carbon chain. No? So depending on the number of carbons, pwede yan maging different types of sugar. No? Discuss natin later. So yan yung isang itsura ng ating sugars or carbohydrates. So you have the aldehyde group here. Sometimes ketone yan. No? And then you have your OHs alternating. Minsan nasa isang side, minsan nasa other side. May meaning yon. And then you have your CH2OH at the end. No? So that's the general motif of sugars. Okay. So you can convert that, no, that is called the Fischer projection. You can actually convert the Fischer projection into other configurations such as the Haworth projection and the chair conformation. Okay, so in the organic chemistry natin last time, alam natin yung chair conformation, no? So ano ba yung chair conformation? That is the most stable conformation for a six-membered ring, no? So, ang example natin doon is the cyclohexane. No? So, di ba maraming maraming conformation. You have the boat, you have the half chair, and then you have the chair conformation. So, the most stable one is the chair conformation. In nature, commonly, makikita natin yung chair conformation. Okay? So, yan. But, again, take note that all this represent the same sugar pa rin naman. Okay? So all these things represent the same sugar. Okay? Ibig sabihin, you can convert the Fischer projection into a Haworth projection and then later on to the chair conformation and vice versa. Okay, you just do the reverse process. You may ask, no, paano siya nagiging circle? Paano siya nagiging ring? Okay, so how come that we have this linear chain of sugar, then later on, mapapansin nyo, naka-ring na siya. So, paano nangyari yon? Okay, one reason is that look at the sugar. It contains an electrophile and a nucleophile. No? So, if you have an electrophile and a nucleophile inside the molecule, pwede magkaroon ng intramolecular reactions. Within the molecule yung reaction. Nadaanan natin yan sa OrgChem last time. Yung cyclization ng chain, no? So, ganun din mangyayari dito. So, yun nga. So, since marami kang electrophile, marami kang nucleophile at may electrophile ka, pwede sila mag-react within itself, no? So, of all the OHs there, yung OH yung ating nucleophile. Remember, nucleophile, maraming electrons, no? Okay, so, oxygen yon in our case. So, of all the oxygens there, sino yung a-attack sa nucleophile? The nucleophile is the carbonyl carbon. Remember that this one has a partial positive charge due to the electron pool of oxygen. So, ito yung electrophile, ito yung mga nucleophile. So, sino dyan yung a-atake? Okay. Commonly, the OH that will attack the electrophile carbonyl carbon is the OH at the penultimate carbon. When you say penultimate carbon, that is the second to the last na carbon. Okay? So for example, ito yung ating carbons, ito yung last, yung CH2OH. Ito yung ultimate. But before that, you have the penultimate carbon, the second carbon to the last. Okay? So yung OH dito, yun yung aatake sa ating carbonyl carbon. And magkakaroon lang ng rearrangement, then we will have the Haworth projection na, which we can later convert into a chair conformation. Okay? So, ganun. So now, let's try to convert this sugar into Haworth and then later on to the chair conformation. Okay? So, if you look at this, no, we have galactose here. Sino yung aatake sa carbonyl carbon? So, sabi ko sa inyo, it is the penultimate carbon na may OH. No? Yung OH sa penultimate carbon, yun yung aatake sa ating electrophile na carbonyl carbon. Okay? So, remember, may lone pair yan. Okay? And that will attack this carbonyl carbon. Ano mangyayari? Magfo-form yan ng ring. Okay? So, magiging ring yan. So, let me show muna yung magbubuong bond. Remember that when your lone pair is donated to the electrophile, magfo-form ng bond. Okay? So, let me show that. Kopyahin ko lang muna ulit lahat. By the way, take note that when the OH is, the lone pair of the OH is bonded to this carbon, mapupush yung electron to the oxygen. So let me show you that. Ilan to? 1, 2, 3, 4. Medyo ihaba ko lang ha. Pero hindi siya ganoon kahaba talaga yung band, no? Kasi papakita ko na dudugtong siya dito. Yan. So yan, magkadugtong na sila. Okay. Take note that may OH pa rin dito, di ba? So kapag dinugtong ko dito yung H, the oxygen will be positively charged, which is unstable, no? Remember that the stability depends on the electronegativity of the atom, no? Sa Lewis structure, no? So kung positive yung formal charge ng oxygen dito, when there is a hydrogen here, unstable yan. So in order to make everything stable, yung hydrogen, instead na dito kay oxygen, ipapasa niya dun sa oxygen na to sa taas, no? Kasi take note that when it accepts the bond, no, ginawa niyang lone pair yan, magiging negatively charged to. No? So to minimize the charges, ipapasa na lang yung hydrogen so that wala tong charge, wala din yung charge. Okay? So, ganun yung mangyayari. So, napapansin nyo na ba yung cyclization natin dito? So, we are actually forming the ring na. Okay? So, ito yung members ng ating ring. 1, 2, 3, 4, 5, 6. No? So, itong chain na to, yung chain na yan, they are naka-ring. No? So, ngayon, mag-numbering tayo. Numbering tayo. So, start tayo dito. 1, 2, 3, 4, 5. So, ang 6 natin is yung oxygen. Okay? So, yun yung 6. Hindi ito. Okay? Hindi iyan. Kasi hindi yan member ng ring eh. Okay? So, yung naka-ring lang yung may numbers. So, yan. Kapag Haworth projection, kailangan mo mag-drawing ng hexagon. Okay? So, mag-draw ka ng hexagon. Haworth. So, you draw a hexagon. Nakapaganyan. Okay. But, yung oxygen, dito siya naka-show. Okay? So, instead na carbon yon, naka-oxygen yun. And let me redraw para mas malinis yung ating hexagon. Okay. Let me redraw that. Oxygen. So, andun talaga yung oxygen parate by default. Okay. So, this is our backbone for the Haworth projection. Okay. And then the numbering of carbon proceeds as this. Ito yung 1, 2, 3, 4, 5, 6 na. So, itong oxygen 6, clockwise yung counting. So, 1, 2, 3, 4, 5, 6. Okay. So, ngayon, remember, may mga substituent yan. You have to show that in your Haworth projection. So, maya na tayo mag-focus sa carbon 1. Punta muna tayo sa carbon 2. This is carbon 1, this is carbon 2. Yung OH natin nasa right side na. Paano mo ilalagay yan sa Haworth projection? Ito yung mnemonic. Let me share you the mnemonic. Ang mnemonic dito is that pag nasa right ng Fischer projection, that is down kapag nasa Haworth. Right down. And then kapag nasa left yan ng Fischer projection, naka-up yan sa Haworth projection. Okay, so again, right down, left up. RDLU. Okay? Right down, right, naka-down sa Haworth. Pag nasa left, naka-up sa Haworth. Okay? So, ibig sabihin, for the second carbon, yung OH na to, since yan ay nasa right, ayan, so since nasa right siya, ibig sabihin, naka-down siya dito. Yung hydrogen, wag na natin ipakita. Okay? So, yung OH lang. Okay? Kasi naka-right siya doon. So, ibig sabihin, naka-down siya sa Haworth. So, let's proceed with the third carbon, which will be here. Ayan, dito siya. So, naka-left yun sa Fischer. So, kailangan naka-up yan sa Haworth. Okay? And then, fourth carbon. Sa fourth carbon, you have the OH here. Ayan, naka-OH yan. Nasa left. So, ibig sabihin, naka-up din yan. Sa fifth carbon, anong gagawin ko dito? Sa CH2OH. Okay. So, generally, pwede yung naka-up, pwede naka-down yan. Okay. Pero, ang gagawin natin, i-up na lang natin yan by convention. Okay. So, you have the CH2OH here. CH2OH. Naka-up yan by convention. Okay. So, yan. So, ngayon, punta tayo sa first carbon. Ito yung problem kasi natin dyan sa first carbon. Naka-up ba or naka-down yung OH dyan? Kasi alam natin, kapag nasa right side ng Fischer, yan ay down. Kapag nasa left side ng Fischer, yan ay naka-up. No? But what we have here is that yung OH, di natin sure kung up siya or down. Bakit kaya ganun? No? Ba't di natin sure kung siya ay naka-up or down or naka-right or left, no, sa Fischer. Kasi ganito yon, no. So if you take a look here sa ating original reaction, yan. So if you take a look at this original reaction here, ano mangyayari? The carbonyl carbon is sp2 hybridized. So ibig sabihin, restricted yung rotation niya. However, as it converts into sp3 hybridized carbon, when the nucleophile attacks onto it, no, ano mangyayari kapag sp3 hybridized siya? So, nagiging free to rotate na siya. Okay? So, ibig sabihin, yung OH natin, it could either be pointing up or pointing down, no? Pwede siyang nasa left, pwede siyang nasa right. Because free yung rotation natin na. Okay? So, magiging free yung rotation. So, that means, dalawa yung possible na Haworth structure for a single Fischer projection. Okay? So, dalawa yung magagawa mo. One is the OH is pointing up. The other is the OH pointing down. Okay? So, para maging mas specific tayo, we will call the first carbon the anomeric carbon. So, siya yung anomeric carbon. Okay? We will call it that. Okay? So, copy ko lang muna yung ibang parts ng ating ring. Okay. So, again, for the anomeric carbon, the first carbon, the OH could either be pointing up or pwede ring naka-down. Okay? So, take note of that. So, mapapansin nyo, we have the same structure, but the arrangement or the spatial arrangement of the substituent, especially on the anomeric carbon, is different. So, ibig sabihin ito ay enantiomer. Okay, so, ano tawag natin sa dalawang enantiomers na to? Okay, so, ganito. Ito yung mnemonic, no? So, let me extend this. Right down alpha, left up beta. Okay? So, ano meaning nun? Okay? So, ganun pa rin naman pag nasa right, naka-down. Pero for the anomeric carbon, pag yun ay naka-down, ibig sabihin that is the alpha isomer. Okay? So, that's the alpha isomer. Okay? Kapag yan ay naka-up, yan ay beta isomer. Okay? So, yan. So, yun ay naka-beta. So, this is important, no? Kasi later on, when we discuss polymers ng sugar, merong alpha, merong beta na sugars. Okay? So, take note that kapag alpha, yung OH sa anomeric carbon is pointing downwards. And kapag beta, the OH of the anomeric carbon is pointing upwards. No? So, kapag alpha, down, beta, up. So, ito yung ating mnemonic, RD alpha, left up beta. Okay. So, those are the Haworth projections we can make from the single Fischer projection. Again, naging dalawa because of the rotation of the sp3 hybridized carbon na when the ring cyclic, pag naging cyclic yung ring natin. Okay. So, ngayon, paano natin ito gagawing chair conformation? Magawa ka muna ng chair conformation. Ito na yung mahirap, mag-drawing ng chair conformation. Kuha ako ng pattern para mas madali. Even until now, na-stress ako sa chair conformation. Ayan. Okay. So we have the chair conformation of a 6-membered ring. By the way, sorry. Oxygen yung nandito, ha. Oxygen to. Let me erase the carbon there. Kailangan oxygen yan. Yan. Palitan natin ng oxygen. Yan, oxygen. Oxygen. Okay. So ngayon, paano natin to ilalagay sa chair conformation? Okay. Kapag yan ay naka-pointing upward, no? Ibig sabihin, naka-up dapat yan sa ating projection. Kapag yan ay naka-pointing downwards, dapat yan ay naka-pointing downwards din. Okay? But take note, for each carbon sa ating chair conformation, they could either be axial or equatorial. Okay? Remember that dito sa carbon na to, axial ang up, ang down ay equatorial. Dito sa carbon na to, dito sa carbon na yon, pati dito sa carbon na yon, ang down ay axial, ang equatorial ay up. Okay? Remember that. Okay? So dito sa carbon na to at ito, up is axial, equatorial is down. For carbons this, and that, ang down ay axial, ang up ay equatorial. Okay? So with that in mind, let us place our substituents. So for the first carbon, the anomeric carbon, the substituent is pointing up. No? So dito sa carbon na to, ang pointing up is the equatorial. Ayan. For the second carbon, OH is pointing down. So for this carbon, remember, ang ating OH dyan ay pointing. Kung naka-down yan, dapat naka-equatorial yan for this OH. No? So, check ko lang yung pattern ko para hindi ako mahirapan mag-drawing. Ayan. So, this is the equatorial. No? For the third carbon, the OH is pointing up. So, ito yung third carbon. So, ang up dito ay equatorial. Okay. For the fourth carbon, the OH is pointing up. So, dito sa fourth carbon na to, ang up ay axial. And for the last carbon, you have the CH2OH, generally pointing upwards. So, dito sa carbon na yon, that can be interpreted as equatorial. Okay? So, that is the chair conformation of our sugar, if that is in beta version. Pag yan ay naka-alpha version, makopyahin mo lang yung iba. CH2OH. Okay. So, ito naman, sa ating anomeric carbon, the OH is pointing downwards. So, ang downwards dito ay axial. Okay. Ayan. So, again, to make things clearer, ganito. Let me draw this. Ayan, bigger. Okay. Ito yung up. Ito yung down. Ito yung up. Ito yung down. Ito yung up. Ito yung down. This is the up. This is the down. Up and down. Okay. So, yan. So, for the chair conformation, this is your carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Okay? So, yan. So, basically, gagawin mo na lang is, i-pattern mo na lang dito sa general structure na ginawa ko yung iyong chair conformation. So, kung sa first carbon yan ay naka-up, then ilagay mo siya dito sa bond na yan. Ayan yan. Sa second carbon, that is down. So, ibig sabihin yan ay naka-equatorial. Ayan. On the third carbon, yan ay naka-up. So, ang up dito ay the equatorial one. On the fourth carbon, yan ay naka-up. So, ito yung up, axial. And on the fifth carbon, generally, yan ay naka-up. So, that is this one, pointing outwards. Okay? So, again, you can use this pattern na lang. Kopyahin nyo na lang yan. So that alam nyo kung saan mo ilalagay yung OH mo, no? Kung yan ba ay naka-axial, naka-equatorial. Kasi per carbon, nag-iiba-iba yon, no? Pwedeng up is, axial. Sometimes yung up ay equatorial. And the reason for that is the Newman conformation. I mean, yeah, the Newman conformation. Yan. Pag kinonvert mo yan into Newman conformation, dun mo makikita yung up-down nila. Okay? So yun, so use that pattern, no, to convert other sugars into convert other sugars into Haworth and chair conformation. Again, take note that dalawa yung mafo-form natin. From your sugar, makakagawa ka ng alpha and beta sa anomeric carbon. Okay? So, since we're talking about the isomerism, tuloy na natin. Okay. So, here are some isomerisms in carbohydrates. First one is the aldose-ketose isomerism. Okay. So, for aldoses, yun ay sugars with aldehydes. No? So, obvious naman. Pag may O's, sugar yon or carbohydrate yan. Aldoses, yun ay sugars or carbohydrates with aldehyde groups. No? So, this one is the aldose. Okay. So, ito yung aldehyde. If you have a ketose, no, ket is the ketone. So, ito yung ketone sa sugar. Okay? So you have an aldose and a ketose sugar. Okay? So, yan. Same formula pa rin yan. Magkaiba lang ng attachment yung atoms natin. Magkaiba yung position ng functional groups. Okay? So, yan. Also, we have, for sugars, the enantiomers D and L. Again, take note that D is dextro rotatory. I'm sobra pala ako ng ro. Okay, so D is dextrorotatory and L is the levorotatory. Okay. So, ano lang yun? It has something to do with the optical activity, no? So, if that's a dextrorotatory, it bends the light in the right hand position. Naka-right side yung kanyang bending. If that's levorotatory, naka-left hand yung bending ng light. No? So, yun. So, it's either clockwise or counterclockwise. No? Okay. So, yun. So, medyo na, alam ko na-discuss natin to sa OrgChem dati. Anyway, then we have the epimers. No? So, epimers, this is diastereomers of the sugars, no? Wherein yung kanilang change is only the position of OH in one of the carbons of your sugar. Okay. So, for example, we have this sugar here, the glucose and the mannose. Pansin mo, yung aldehyde, same lang for the the second carbon. Ayan. Papansin mo, yung OH dito, nasa right side for the glucose. For the mannose, nasa left side ang OH. No? So that means they are C2 epimer, carbon 2 epimer. Doon lang sila nagkaiba. But for the others ay same lang yung position nila. Okay? So maraming isomers yung ating sugar, no? Kasi pagbali-balik ta rin mo lang yung OH, may magagawa ka ng other version niya. Okay? So yan. So that means that mannose is the C2 epimer of glucose. Paano mo malalaman kung anong carbon epimer yan? So, check mo lang kung saan sila nagka-switch. Okay. So, what if dito sila nag-switch? Ito, pati ayun. So, you can call that the C5 epimer. Bakit C5? Kasi pang-limang carbon siya nagkaroon ng changes. Kung sa fourth carbon yan, edi C4 epimer. Kung sa third carbon yan, edi C3 epimer. Doon lang sila nag-switch. But the others should be the same. Another isomerism is the Pyranose and the Furanose. Okay. So, pag Pyranose, yan ay 6-membered ring, which we just shown sa ating Haworth projection. Okay. Pag Furanose, ito ay 5-membered ring. Okay. Ang aldoses ay nakaka-produce ng Pyranoses. So, pag aldose sugar ka, Pyranose yung inyong ring structure. Kasi magiging 6-membered yun, di ba? Remember, kapag niring natin yun, 6 yun, di ba? So, aldose yun. Pag ketoses ka naman, you have the furanose version. Okay? Kasi nangyayari for ketoses, nasa second carbon yung carbonyl, no? So, kapag sincyclic mo yon sa yung penultimate OH, kakabit siya sa second carbonyl carbon, magiging 5-membered na lang yung ring natin. Okay? So you will form the Furanose one. Same rules apply, no? Right down alpha, left up beta. Okay. As you can see, the OH here is down, so it is alpha. Here is down din siya, so this is alpha. Siguro you may ask, no, paano namin malalaman if yan ay D or L? Okay. So malalaman mo kung yan ay D or L, nakalimutan ko pala daanan. Kapag D, naka-right side yung OH sa penultimate carbon. So this should be the penultimate carbon. I mean, the penultimate OH. Pag nasa right side, naka-D, dextrorotatory. Kaya right hand yung rotation ng ating light. So kapag yan ay nasa left side, yan ay levorotatory or L. And then, of course, nakwentuhan na natin to. We have the alpha and beta anomers, no? So remember that the first carbon is the anomeric carbon, no, in the Haworth projection. So if that's pointing up, that's beta. And if that's pointing down, that's alpha. Okay. So now let's talk about, let's go back with the general idea of carbohydrates or sugars. Maraming versions yan, no? We have the monosaccharide, the oligosaccharide, or disaccharide minsan kapag dalawa lang. And then we have the polysaccharide, no? So those prefixes, the mono, di, oligo, and the poly, just refers to the number of sugar molecules in your chain, no? In your polymer, no? No, kapag isa lang yung inyong isa lang yung sugar mo dyan, ibig sabihin monosaccharide yan. If you have 2 or more, at least 20, you have the oligosaccharide. And if you have more than 20, yan ay polysaccharide na. Okay? So yan. So ano lang yung difference ng mga yan? Number of sugars. No? When we say saccharide pala, that is sugar in Greek. Okay. So for monosaccharides, these are the simplest sugar. Okay, so this is the simplest sugar. And siya ay, ang pinaka-abundant na monosaccharide is apparently the glucose. No? Present yan sa halos lahat. No? Even yung kahoy natin, may glucose yan. Kaya nga sabi ko sa inyo, kung kaya nyo ma-digest yung kahoy, magkaka-energy kayo. But apparently, we cannot. No? Okay? Ayan, nagbalik ulit siya. So, we have the aldose and the ketose. Okay. Anyway, so these are the monomers, no? So, for aldoses, no? Ibig sabihin, aldehydes, no? So, ito yung ilang example. So, for 3-carbon, we only have the glyceraldehyde. Okay. Then, for 4-carbon chain, you have the erythrose and the threose. Okay. So, wag niyo nakabisaduhin to. Pwede nyo naman tignan na lang yung sugar. Okay, so don't memorize this na lang. And then if we have 5-carbon chain, the dami niya, magdo-double yan. So we have the ribose, which is used in ribose sugars. Okay. The arabinose, the xylose, and the lysose. And then if you have a 6-carbon chain, magdo-double yan to have ito. Then, so you have the allose, altrose, glucose, mannose, gulose, idose, galactose, and talose. No? Okay? So, mabansin nyo, when you add carbons, nagiging times 2 yung dami nila. No? For example, ito, 3-carbon. Dagdag ka ng isa dyan, magiging times 2 yung dami niya. Bakit? Kasi because of the epimer. Okay? So, because of the position of the OH, dun sila nagkakaiba lang. For example, ito, OH nasa right side, ito nasa left side. So, that means erythrose is the epimer, C2 epimer of threose. Pag nagdagdag ka uli ng carbon dyan, okay, pag nagdagdag ka ng carbon dyan, ito pwede maging left-right. So, ito yung dalawa yan. Dito, pwede maging left-right din yung OH. So, itong dalawa yan. Then, the same thing will happen for all of them, producing many types of monosaccharides. So, ito yung aldoses natin. Aldehyde yung kanilang first carbon. If you have ketose, ito yung tsura nila. You have a ketone. Same concept pa rin. So, for 3 carbons, ito lang siya. For 4 carbons, when you add carbon, after yun ng carbonyl parate. No? So always after the carbonyl, dun ka nag-add ng carbon. However, isa lang magagawa mo dyan. Okay. So isa lang yan. Then pag 5 na yung carbon mo, dun lang magkakaroon ng times 2 yung dami natin because of the epimers. And then for 6 carbons, ayan. So ito yung mga versions nila. So yung magkakaiba pangalan, no? So we have the dihydroxyacetone, the erythrulose, ribulose, cellulose, psychose, fructose, sorbose, tagatose. No? So yan yung mga sugar natin. Unti lang yung ketoses natin. But for the aldoses, marami yan. Okay? Okay, so again, don't remember this. You can check them naman always. For oligosaccharides, ito na yung mga more than one, syempre, but not so many. So, basically, what we're doing here is pagdudugtong-dugtungin mo na yung sugars mo. Okay? So, yan. So, commonly, disaccharide yung marami sa paligid natin. Ibig sabihin, ang dalawang sugar na pinagdugtong. And then, the connection between two sugars are called glycosidic bonds na. So again, may additional tayo na term for the connection of two sugars. Pag yan ay proteins, tawag natin sa connection ng dalawang amino acid ay peptide bonds. Pag yan ay nucleic acid, ang tawag natin sa connection nila ay phosphodiester bond. Kapag yan ay sugar, ang connection ng dalawang monomers ng sugar, which is the monosaccharide, ay glycosidic bond. No? So, yan. So, glycosidic bonds are products of the condensation of two sugars. No? So, paano yung nangyayari? Ganito lang. So, when your two sugars condense, what will happen is that yung H and OH nila, may lalabas yung sa reaction, and then this oxygen will attach to this carbon. Okay? So, yan. So, yan. So, magkakaroon ng attack dito. This can be protonated. And this can be protonated, so this can be a leaving group. Then, if you have a carbonyl carbocation na dito, the oxygen can attack that, no? So they will condense into maltose. Disaccharide. Okay. So yun. So yan, so that's the condensation, and that's reversible. No, if you hydrolyze this, you will form the two monosaccharides again. Okay. So sa ating disaccharides, there are common disaccharides that we encounter every day. So you have to take note of this. Okay. So you have lactose, no? So kapag wala ka ng lactase, the enzyme that destroys this, giging lactose intolerant ka. Sasakit tiyan mo parati sa dairy products. So lactose is very abundant in dairy products, no? So ang ating lactose, yan ay gawa sa galactose and glucose, no? Okay. So you have a galactose connected with the glucose. What is this? Beta 1,4. Ano meaning nun? Okay. So meaning ng beta 1,4 is ganito. Okay. Yung galactose ay naka-beta, yung first carbon. So ibig sabihin naka-pointing upwards yung OH niyan. Then yung 1,4 meaning nun, yung first carbon dito, madudugtong yan sa fourth carbon ng next sugar. Okay. So beta 1,4, ibig sabihin pointing upward yung first carbon ng galactose here. And that will be connected to the oxygen of the glucose down here. So first carbon to fourth carbon. Okay, remember, 1, 2, 3, 4, yan yung numbering natin. First carbon, fourth carbon. So since this is nakataas, that's beta. So that's lactose. We have the galactose, beta 1,4 glucose. Okay. Then we have the sucrose, which is the table sugar. Okay. So the table sugar is a combination of glucose and fructose. No? A 6-membered ring and a 5-membered ring. No? May Pyranose and Furanose ka. Okay. So ito yung complete name ng sucrose. Glucose alpha 1,2 fructose. Ano meaning na? Ganito lang. Okay. So you have your glucose here on the left side. Alpha meaning the OH there is pointing downward. So, naka-down yung ating bond. Okay? 1, 2, ibig sabihin yan, the connection between the glucose and the fructose is sa carbon 1 and sa carbon 2. So, dun sila magdudugtong. Okay? So, yan. Then we have the maltose that is two glucose molecules connected to one another. And so we have maltose, glucose alpha 1,4-glucose. Alpha meaning naka-down yung OH. Okay? And connected yung first carbon to the fourth carbon of the other sugar. And we have the cellubiose. Okay? So we have the cellubiose, which is nasa cellulose. Okay, kaya cellubiose pangalan niya. So that's glucose na naka-beta 1,4 position naman. So for cellubiose, the glucose here should be beta. Okay. Kaya nakapaganon yung bond. Naka-up kasi yung OH dito. Kaya pag ganyan yung bond, pa-diagonal. So this is the first carbon connected to the 4 carbon of the other glucose. Also, you should take note that ito, tignan nyo to. If you have your second sugar, tignan nyo yung second sugar. If the anomeric carbon has HOH, no, may hydrogen and may OH done, you have a free hydroxyl group on the anomeric carbon of the second sugar, then that is reducing sugar. Reducing sugar to. Okay? Otherwise, kung wala yung H2O sa anomeric carbon, then hindi yan reducing sugar. Okay? So that characteristic of sugar being reducing sugar or not, but yun yung mga ginagamit namin to classify the sugars, no? To compare them from one another. Okay? So again, take note that if the second sugar has a free OH on the anomeric carbon, then that is a reducing sugar. Okay. So last one, we have the polysaccharides. Okay. So when you say polysaccharides, this is poly, marami. More than 20 sugars na yung pinagdudugtong-dugtong natin. We also call polysaccharides as the glycans. No? So ibig sabihin yung kanina sa peptido glycan natin, that is the combination of peptide chain and the glycan, which is the polysaccharide. Yun sa gram-positive and gram-negative bacteria. Okay. So, yan. So this is composed of combinations of more than 20 sugars. No? Okay. So varying yung varying yung chain niya. No? So pwede magkaroon ng cross-linking. Okay. So ibig sabihin pwede magdugtong-dugtong yung maraming sugars. So and some of the examples of polysaccharides are the starch and the cellulose. No? Okay. So ito yung ating versions of the polysaccharides. So they could be unbranched homopolysaccharide. Ibig sabihin same sugar lang and di isa lang yung kanilang line. If you have a branched homopolysaccharide, ibig sabihin yan, you have the same sugar molecules pero may branching na. So hindi lang siya straight lines. Pwedeng meron dito, pwedeng meron doon, no? So pwede may branches siya. If you have a heteropolysaccharide, so this is more than two sugars ang gamit niya. So pwedeng unbranch yan, straight line, and pwede ring branch, no? So pwede may branching. Okay? So let's look at one example. So we have the starch, no? We know that starch is the energy storage form of plants, no? Kaya yung mga patatas, yung starch doon, yun ay puro sugar yun. Okay. Even yung ating mga prutas, no? Yung starch sa, let's say, sa apple, yun yung storage form ng halaman. Okay? So that's the energy reservoir of plants. Okay? And gawa siya sa dalawang uri ng polysaccharide. We have the amylose and the amylopectin. Okay? So when you say amylose, this is a polymer of sugar molecules, especially glucose, connected via alpha 1,4 linkage. No? So you have a sugar, glucose na magkakadugtong and they are interconnected via alpha 1,4 linkages. So yung mga OH natin nasa baba. Okay. amylose, alpha 1,4 linkage yung ating glucose. When we have amylopectin naman, so ito ay branch polymer na. Okay? So ang connection dito is other than you have the alpha 1,4, you also have the alpha 1,6. Okay? So hindi lang alpha 1,4 siya, meron ding alpha 1,6. For example, you have here the alpha 1,4 for this sugar and that sugar. So ito, pwede rin siya mag-branch doon. Na? So ito yung first carbon, ito yung 6 carbon of the other sugar. So this is the alpha 1,6 glycosidic bond. Okay? So multiple. So ibig sabihin, pag amylose yan ay unbranched. Pag amylopectin, since you have two connections ng sugar, alpha 1,4 and alpha 1,6, then yan ay branch, no? Ano nangyayari kapag nagiging branch yung ating saccharides, no? Nagiging malagkit sila. Okay. So nagiging malagkit sila because yan, branch yung molecules, no? Nagiging malagkit sila. So ito yung ating starch, no? So that is a combination of amylose and amylopectin. Yan. So yan yung kanilang general format. So they are intertwined to one another. Next one, we have the glycogen, which is the storage form of animals, no? So tayo yun. May mga taba-taba tayo, mga bilbil natin. Gawa yan sa lipids and sometimes glycogen then. Okay. So this is the storage form of animals, no? Of animal cell. And this is a polymer composed of alpha 1 for connection of glucose with alpha 1,6 branches then. Okay. So medyo similar siya with the amylopectin. Alpha 1,4 and alpha 1,6 now. But this time nasa animal cell na tayo. Okay? So yan. So yan, pag nagkumpul-kumpul yan, maging malaking mass yan ng fat sa ating tiyan, no? Sa ating bilbil, no? So if you want to remove that, no? Ang ginagawa ng body natin, kunwari hindi ka kumakain, nag-diet ka, uubusin niya yung inyong glycogen using an enzyme called the glycogen phosphorylase, no? So puputulin niya yan. Okay. So itong enzyme na to, glycogen phosphorylase, yan yung ginagamit ng body natin to cut the fats, no? I mean the glycogens, no? Yan. Next type of polysaccharide, we have the cellulose, no? So again, cellulose, ito yung nasa plants natin. Ito ay composed of sugar, the same sugar, glucose pa rin. However, the connection is beta 1,4. Okay? So compare that with the alpha 1,4 sa amylose, no? Okay, so sa amylose, alpha 1,4 ang glucose. Pag cellulose, beta 1,4. Okay? So yan lang yung difference nila. Siguro you may ask, no? Bakit tayo pwede natin kainin yung starch, no? Yung mga alpha 1,4, alpha 1,6, no? Pero yung ating cellulose, di natin makain, beta 1,4. Ganito kasi. Ano yung identity ng enzyme sa ating laway, no? So alam natin yung enzyme sa ating laway is called the alpha amylase. Enzyme yun ng amylase, ng amylose, no? Kaya nga amylase pangalan niya. But because it is alpha, it can only cut the alpha bonds, no? Amylase, enzyme yun ng amylose. Alpha, kasi it can cut the alpha bonds only. Ang problema, sa cell wall ng halaman, naka-beta siya. So, ibig sabihin, kapag kumain ka ng halaman, hindi niya mabe-breakdown yung bonds kasi, again, specific siya for amylase only and alpha 1,4 dapat yung linkage. But for cellulose, hindi eh. No, beta 1,4 yung linkage. So, hindi niya makakat yan. That's why kapag kumain ka ng mais, yung starch sa loob ng mais, mauubos yon, matutunaw yon. Pero yung cell wall niya, yung film niya, yung coating ng mais mismo, buo yun na malalabas, no? Kunwari, kumain ka rin ng gulay, no? So, yung starch ng gulay, mauubos yon, but yung cellulose niya, hindi niya mauubos yun. Kasi wala tayong beta 1,4 na enzyme. Wala tayong beta cellulase. So yan. So ano, reason, ano ang dahilan nun? Wala, ganun talaga, no? So ang tao ay designed for amylase consumption only, amylose consumption. Wala tayong cellulase, no? Kasi imagine nyo, kung may cellulase tayo, edi para na tayong insekto, no? Kumakain na tayo ng kahoy ngayon, no? Which is okay sana, no? Kaso weird, no? Imagine nyo, pagpunta ka ng restaurant, no? O, ito kahoy, 5,000. May gastos ka ng malaki para nun sa kahoy. Medyo weird yun na pag ganun yung nangyari. So siguro designed lang talaga tayo to be like this, no? That we are designed to just eat the alpha glycosidic bonds instead of the beta glycosidic bonds, no? Okay. So yun lang. Pero imagine the world, no, na tayo ay naka-beta, may beta cellulase tayo. Ano kaya mangyayari, no? Walang world hunger na, no? Kasi kahit kumain ka ng kahoy, busog ka. But that's not the case, no? Siguro kung makakagawa kayo, no, in the future, kunwari mag-research kayo, gawa kayo ng enzyme na cellulase na good for people. Ayun, baka matanggal yung world hunger. But I don't think that's possible, no? Kasi kung possible yan, hindi sana matagal na na-address yung issue. But hindi pa rin. So siguro impossible yan. Okay, so ito yung itsura ng cellulose natin. As you can see, may OHs yung ating mga sugars, so they can form hydrogen bond na, making it much stronger. So another one is the chitin. Okay. So chitin, ito yung mga nasa crustaceans naman, no? Yung mga nasa crabs, no? So yan. So ang kanilang coating ay gawa sa chitin. So for example, yung crabs, no, yung shell na yun, chitin yun. Okay. So ang chitin naman is gawa siya sa different form of sugar, no. So the monomer there is not a simple sugar, rather it is a modified sugar called the N-acetyl beta D-glucosamine. Okay. And the connection between chitin is that yung ating mga N-acetyl beta D-glucosamine, they are connected via beta 1,4 glycosidic bonds. Okay. So, modified yung kanyang sugar. Okay, so now let's talk about glycoconjugates. No? When you say conjugates, other versions of sugars. So, tatlo yan. We have the proteoglycans, the glycoproteins, and the glycolipids. No? Basically, ito yung combination na ng proteins or lipids and sugars. No? So let's talk about proteoglycans. So from the word itself, prote, ibig sabihin protein, and then the glycan meaning sugar. Okay. So for proteoglycans, this is a combination of sugar, carbohydrates, and proteins. No? So ito yung nakikita natin sa mga cartilage natin, sa mga connective tissues such as cartilage, no, mga tendons, ayan. So they have proteoglycans. So pwede mo kainin yung tendon ng manok, no. Okay. Although ako, I don't eat chicken feet, no. Kasi they are rich in carbohydrates and proteins then. Okay. So healthy yun. So for glycoproteins naman, so you still have the protein and the sugar. Okay. So yan. However, ang bonding ay different. No? Okay. So iba yung bonding nila dito. Okay. So ang bonding ng sa glycoprotein, the bonding is sugar, connected siya sa oxygen ng ating serine, or pwede siyang connected sa nitrogen ng ating asparagine. So, dun lang sila nagko-connect. But for proteoglycans, kahit saan. No? Okay. So, saan ito ginagamit? Ito ay ginagamit sa mga enzymes din. Glycoproteins. The next one, we have the glycolipids. No? So, these are lipids connected with sugars. No? And ano purpose niyan? So, pwede ito gamitin sa cell signaling, just like what I mentioned earlier. And pwede rin ito na maging essential or integral part ng cell membrane. No? Medyo malapit na tayo matapos. We have the reactions of monosaccharide. So, our monosaccharides can be oxidized using the three different acids that we have here, the aldonic, the uronic, and the aldaric acid. No? So, if you have a sugar and you react it with aldonic acid, no, magbabago yung anyong carbonyl group doon. Usually, yung carbonyl group yung nagbabago. Okay? So, yan. So, for example, we have this one. We have the glucose. If we oxidize the first carbon, then magiging gluconic acid yan, which is aldonic acid. Okay? If you oxidize the ultimate carbon, last carbon, we will have the uronic acid, the glucuronic acid. And then if you oxidize both sides, you will have the aldaric acid. Okay. So may carboxylic groups yung tig isang dulo nila, the first and the last carbon. Okay. So again, take note that for oxidation of the first carbon from aldehyde, magiging carboxylic acid. Ang tawag dito ay aldonic acids. C1 oxidation. If you have C6 oxidation, you have the uronic acids. Okay, carboxylic acid yan. But if you have C1 and C6 oxidation, you have the aldaric acid. You may ask siguro, anong oxidizing agent dyan? Typically, nitric acid ang gamit. Okay. So, ang general acid na ginagamit dito is the nitric acid. Okay. So, yan. And then we have the reduction. Okay. So, pag nag-reduce tayo, typically, ang aatakihin mo dyan is the carbonyl carbon because the oxidation number of carbonyl carbon is high. So, gagawin mo, tatanggalin mo yung double bond. Therefore, you reduce it into simple alkane. Magiging alcohol na lang siya from aldehyde to alcohol. Okay? So, yan. So, ang tawag natin for aldoses that are reduced, yung sabihin yung ating aldehyde group doon ay na-convert into alcohol na lang, ay alditols. Okay? So, anong reagent ginagamit dito? Typically, mga reducing agents, We can use, H2 platinum, right? And other reducing agents. For chain lengthening, this is called the Killiani-Fischer synthesis, okay? So, gagawin mo dito, you react your sugar with hydrocyanic acid, then later on, you reduce it, And then, when you reduce it, you add water para maging carbonyl na siya ulit. Okay? So what will happen here is ganito lang. I will not go into much details na lang. Okay? So itong CN natin, okay? Yung nitrogen, this will serve as your nucleophile and your carbon will serve as your electrophile. So the nitrogen will attack this. Then maliliberate yung water. Kakaroon ng rearrangement. So mangyari, itong aldehyde group mo magiging cyano group na. Ayun, CN na lang siya. Then when you reduce that, it will convert into this one. Okay. And then when you add water, okay. So when you add water, magkakaroon lang yan ng substitution, giving us the aldehyde. Ano yung takeaway natin dito? Takeaway natin dito is that madadagdagan tayo ng oxygen bonds dito. So, madadagdagan tayo ng carbon. So, ibig sabihin, mag-ilalengthen yung ating chain. Okay? So, ayan lang. So, ano pala dito? By the way, epimers yung magagawa natin. Okay? So, epimers magagawa natin kasi the OHs na nadagdag natin will be either positioned left or right. Okay? So, ayan. So let's have some example reaction here. Predict the products of the following Killiani-Fischer synthesis. Okay. So basically, ito lang idea sa Killiani-Fischer synthesis. You have this structure. Madadagdagan lang siya ng carbon dito. So yung OH pwede andyan or pwede rin naman ng dito. Okay. So hanapin na lang natin yung molecule that has the same structure dito sa baba. So same na tatlo yung OH sa right side. Then yung isa nasa left, yung isa nasa right yung OH for the added carbon. Okay, so ito, tatlo yung OH sa gilid. Okay. Ito rin tatlo yung OH sa gilid. So, ito yung mga possible products ng Killiani-Fischer synthesis natin. Ang 2 hindi, 3 hindi, 4 hindi, no? Kasi mapapansin mo, yung OHs should be on the same side. Ang magbabago lang is yung added carbon mo. Okay? Doon lang siya magiging epimer. Okay? So, doon lang. Either nasa right or nasa left. So, ganun yung pag-identify lang ng product sa Killiani-Fischer synthesis. Again, hindi na hindi na tayo magre-reaction mechanism. So, ano lang, identification lang. Quick identification. And then we have the chain shortening, the vol degradation. So, for the vol degradation, basically you convert your carbonyl group into something that is a good living group na. And then once na mag-live na siya, iigsi na yung carbon chain mo. Okay? So this is the typical flow of the reaction. You have your sugar. You react it with a strong base. Okay? So you react it with a strong base. So may ma-form ta, an oxime. And then you react it with ACO. AC means acetate oxide. No? So you react it with acetate oxide. So this will be reduced. No? So you will have your cyanide, and then you react it with another strong base. No? So ma-convert yan into aldehydes again. So again, yung mechanism, hindi na natin siya pansinin kasi tapos na tayo sa OrgChem. But the idea is that for chain shortening, umiigsi yung ating chain. Typically, ang natatanggal ay ito, the second carbon. So, yan yung nawawala. Okay? So, ibig sabihin, mariretain ito. Mariretain yung parts niya. But for the second carbon, mawawala siya. Naging aldehyde siya. Another thing is the rough degradation. So, similar idea. Okay? Similar idea. However, instead of converting your sugar into cyanohydrine, what we do in rough degradation is you convert it into carboxylic acid, no? Which is later reduced into aldehyde. Okay? So, yeah. So, medyo same lang din yung idea dito. Yung second carbon yung nabubura parate. No? Matitira yung 3 carbons. Yan. So, matitira yung last 3 carbons. But yung second carbon, yun yung nawawala. Okay. So basically, let's try this reaction. We have the rough degradation of ribose. Ano yung magiging product? So, di na natin papakita yung mechanism, just the final answer. Sabi ko sa inyo, yung second carbon yung mawawala. No? Ito yung mawawala. No? Okay, so, you just copy everything. Just copy everything, pero mawawala na yung second carbon. Okay, so mawawala lang yun. Second carbon always on target. So this is your product. Ano yung name niya? Just look up na lang sa table. Okay. Hindi ko rin kabisado kasi hindi naman siya kailangan kabisado. Ito pala, erythrose. So the product is erythrose. So you can see, yan, matitira na lang itong dalawang OH doon. Yung second carbon always mawawala. Next one is the ozone formation. So sa ozone formation, ginagawa dito is that you convert your aldoses and ketoses into the same compound. No? So yeah, so you just convert them into the same compound. No? And then from their reaction, you can actually compare whether they are of the same sugar groups. Okay. So kunwari ito, let's try this. What monosaccharide form? The same ozone as the ribose. Okay. So, ano yung kailangan mo i-observe dito sa ozone formation? Mangyayari sa ozone formation is that yung until second carbon, magbabago yan. Okay. Pero yung other carbons sa baba, hindi. Okay. So, that means, kung gusto mo malaman kung sino may same ozone formation, you just remove this from the group. Okay. Remove mo lang yan from the group. So you just remove this from the group and then observe if they have the same connections dito sa dalawang carbon sa baba. So for ribose, yung OH dalawa na nasa kanan. So the correct answer here is the Arabinose. Kasi yung OH niya rin na dalawa sa baba ay naka-right side. Ang xylose and lysose hindi. No? Okay. So again, yun lang yung idea dito. Just look at the sugar structure. Always ang carbon 1 and carbon 2 yung nagbabago, but the rest is not, no? Hindi sila nagbabago. Another reaction of our monosaccharides is the ester formation. Okay, so basically, you just convert your alcohols into esters, no? And that can be done using acetate oxide, no? Okay, so yan lang. So lahat ng OHs mo magiging ester. If you want to convert that into ethers naman, then you need something like methyl iodide, no? And react it with silver 1 oxide. So yan lang. So magiging ether yan. By the way, kapag sinabing ester, ganito yung itsura, ha? And so you have your double bond O, then O. So yan. So ganun kasi mangyayari dyan, no? This is then ester. Kapag ano naman, ether, ano lang yan, OR then R. For phosphorylation, okay, so ito yung nangyayari sa glycolysis. So what happens here is that the 6 OH na yung nasa pinakadulo, yung OH na yun that is being converted into phosphate groups. No? So yun yung phosphorylation, and this will actually kickstart the glycolysis process. So mabilisan natin to discuss for the last chapter. Okay, so this is the mechanism for the conversion of glucose reacting with adenosine triphosphate to produce glucose 6-phosphate, which will be used in glycolysis. At the last one, let's identify if this is a reducing sugar. Again, an idea natin kanina is that the anomeric carbon should have a free OH. No? When it has a free OH, it is a reducing sugar. This is your anomeric carbon, and kailangan may OH na nakadugtong dyan. Ano yan eh? OCH3 yan eh, methoxy group yan eh. So since this is not OH, then this is not a reducing sugar. Not a reducing sugar. Okay. So that's all for sugars. No? That's all for carbohydrates. Stay tuned for the next part, which is on the metabolism. Okay. So thank you for watching this video, and hopefully may natutunan. If you have questions, don't hesitate to ask me. Okay. So thank you, and I'll see you again in the next video.